Imagine a tool so powerful that it could rewrite our genetic code. A tool that could one day cure cancer, eradicate genetic disorders, and even reshape the plants we grow and the food we eat. Sounds like science fiction, doesn't it? But this isn't some far-off futuristic idea. It's happening right now, all thanks to advancements in biotechnology, especially a groundbreaking technique called CRISPR. So what exactly is CRISPR? Let's break it down. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is essentially a high-precision DNA editor. Think of it as a pair of molecular scissors paired with GPS. Scientists can cut specific parts of an organism's DNA and paste in new sequences exactly where they want. It's fast, accurate, and, in comparison to older genetic modification techniques, surprisingly easy to use. The possibilities here are enormous. Imagine a farmer growing crops that thrive in drought conditions or resist pests without the need for chemical pesticides. Envision doctors using CRISPR to target cancer cells and destroy them without harming healthy tissue. Parents of children with rare genetic disorders may one day see those diseases vanish, not only for their child, but for all future generations. And this revolution is already making headlines. Scientists are using CRISPR to create malaria-resistant mosquitoes, potentially saving millions from suffering and death. Researchers are experimenting with CRISPR-based therapies for diseases like sickle cell anemia and muscular dystrophy, afflictions that were once thought incurable. CRISPR is a revolution in how we see life, at its most fundamental level. But as promising as the technology is, we mustn't be blinded by its potential. Because with great power comes great responsibility, and CRISPR is no exception. Biotechnology, like CRISPR, touches the very blueprint of life itself. And tampering with that blueprint can sometimes lead to consequences we don't fully understand. Let's start with unintended genetic mutations. DNA is incredibly complex, and isolating one gene can have ripple effects that influence others in ways we don't anticipate. For instance, by creating pest-resistant crops, could we accidentally produce plants that harm beneficial insects? Could changes to human DNA intended to cure a disease create new vulnerabilities? The truth is, we're exploring uncharted territory, where every edit matters, but we don't yet have a complete map. Then there's the question of ethics. Picture a future where parents decide the traits of their unborn children: intelligence, eye color, athletic performance, even personality. Designer babies might sound appealing to some, but at what cost to society? Who gets access to these enhancements? Would it create a world where the wealthy reap the benefits of perfection, while the less fortunate are left behind? And how would it change how we see each other, as unique individuals or as customized products of genetic engineering? These aren't abstract debates, they're real ethical dilemmas we're already beginning to face. But perhaps one of the most pressing concerns is security, biological security. CRISPR and similar tools are becoming more accessible, and with that accessibility comes risk. An accidental release of a harmful genetically modified organism could disrupt ecosystems in ways we can't reverse. Worse, malicious actors, such as bioterrorists, could weaponize biotechnology to engineer pathogens capable of causing a pandemic far deadlier than anything we've ever known. The thought alone is terrifying, and it places a heavy burden on governments, scientists, and regulators to act responsibly. So where does that leave us? CRISPR is, without a doubt, one of the most significant breakthroughs in modern science. Its potential to do good is staggering, but so are the risks if it's misused or mishandled. That's why collaboration is so vital. Scientists, ethicists, and policymakers must work together to set boundaries, establish strong oversight, and carefully weigh the risks and rewards of each application of genetic editing. CRISPR holds a mirror to our society. It asks us to reflect not just on what we can do, but on what we should do. It challenges us to consider how we balance innovation and responsibility, promise and precaution. The future of biotechnology is unfolding right before our eyes. Whether it becomes a tool for progress or peril will depend on the choices we make today. The question is, are we ready to make them?